So we're on the last little bit of the cells, right, of the innate. I kind of debated back and forth on whether I wanted to talk about some stuff as it related to adaptive just yet, and we learn from repetition, most of us, right? So I think it'd be better if I introduce it now and then say it again and say it again, like gram-negative and gram-positive, right? The more I repeat it, the more likely you are to remember it. So I don't have a lot of slides for this. I just posted them right now. Again, I posted them right before because I really didn't want you guys to see them beforehand. I want you guys to pay attention. But you have access to them after, or right now. So I want to review a little bit, right? Because one of the things I asked you guys to try and do for homework, it was to try and fill in the diagrams, right, that I posted. One of them we went over. One of them we're going to go over today. But don't look at your notes right now. I want you to see how much you remember from doing that. Okay? So you need to be able to describe the steps involved in the process of phagocytosis and be able to describe respiratory burst and its effects on microbes. Right? And so for the first one, right, we have this diagram. And so I blocked out some of the key components that you should know. So, and I numbered them because this one is, down here instead of up there, so make sure I go in order, the order that I wanted to go in. So what cells have the ability to do phagocytosis? That's labeled right here in our diagram, but see if you can remember. What are the names, specific names of 2 white blood cells that can do phagocytosis that we've learned about? Neutrophils is one of them. Monocytes when they're in your blood, but what are they called when they're in your tissues? They're called macrophages, right? So these are your phagocytic cells. Macrophages are in your tissues all the time, but they'll get called in during inflammation. And then, of course, the first ones on the scene are going to be the neutrophils. And they're literally going to devour and destroy the microbes in that area, whether it be a virus, a bacteria, right? If it's small enough that it can engulf it, it will. So how does it know, the very first step, how does it know that that's the bad guy? What has marked Yeah, I screwed up what I wanted to write there. What marks the cells for destruction? Little sensors. Be more specific. So what on the surface of this cell is going to tell, right, of this microbe, is going to tell the neutrophil or the macrophage that this is a bad guy? Say again? Pattern recognition receptors. So our cells have recognition receptors, right? So they may be recognizing patterns on the microbe itself. Okay? What does our body actually make that will mark them for destruction, that will actually bind to the cells? We make antibodies. That's one thing. What's something that's non-specific that's a product of your innate immune system? They're called opsonins. Okay, give me an example of a specific opsonin. Something maybe that complement protein, a complement protein. C3b, right? Can you guys think of something else that our body produces that'll bind to their surface and tag them for destruction? Yep, mannose, binding lictins, reactive C-reactive protein, excuse me, right? So these are all opsonins, right? They're actually produced by our body to recognize specific patterns, right, or specific things on the microbes. The next thing is, inside these cells, they have structures that contain enzymes, specifically the enzyme lysozyme. What do we call those structures? They're important for phagocytosis. In addition to lysozyme, they contain defensins, other digestive proteins. What do you call them? You need to know this, guys. Lysozyme is a very big clue. It's just a lysosome. Right? But what a lysosome is, right, is this membrane-encased vesicle inside these phagocytic cells, these macrophages and these neutrophils. And they contain things like lysozyme, which, what does that do? What does lysozyme do? What on the bacteria does it eat or digest? Peptidoglycan, right? It destroys peptidoglycan. So it's most effective against gram-positive or gram-negative? Gram-positive, why? There's more peptidoglycan. Why does it not work against gram-negatives? They have a little layer of peptidoglycan, but what do they have that surrounds the peptidoglycan and stops the enzyme from getting to the peptidoglycan? You got it, Christian. Right. What do we call that membrane, though? That one has a specific name. That's the outer membrane, right? That's the outer membrane. So lysozyme is only one of the things that's in the lysosomes, but they're named after that, right? So they contain defensins, which are proteins that will insert into the membranes, other digestive enzymes. So the next step is actually, once the receptor binds to the opsonin or some other pattern recognition molecule on its surface, then it's going to actually surround it with its membrane, right? And that forms another vesicle that has a specific name. What do we call that vesicle? So this is when they've actually engulfed the microbe. You're right. You got it. Almost, I think. No, it's not a phagolysosome yet. It's just a phagosome, right? And actually the answer is right there too. I didn't cover it up, right? This forms the phagosome. Now, they're showing hydrogen ions going in because one of the first things that will enter into that cell are hydrogen ions. What does that do to the inside of that vesicle? What are hydrogen ions? No. Hydrogen ions are protons. And we measure the amount of these in a solution by measuring pH, right? So if we're adding hydrogen ions, what are we doing to the pH of this vesicle? We're making it acidic, right? Acidification is one of the first things that happens, right? By adding hydrogen ions. And this will potentially deactivate, right? May even kill the microbe that we've ingested. But that might not be enough. So the next thing that needs to happen is that notice the lysosomes fume with the phagosome. So guess what we do with the name? That vesicle now gets called a phagolysosome. We fuse the two names, right? So now it's a phagolysosome. So this is now when we're digesting it with the digestive enzymes that are found in the lysosome. The other thing that we will do is oxidative burst. So what is oxidative burst? What is that? You need to know it. It's one of our objectives. So why is it called oxidative? What are we referring to when we say oxidative? We're talking about oxygen, right? Are we talking about nice oxygen? Friendly? We need it for respiration? No. In this case, can you guys think of a toxic form of oxygen? You might even have in your medicine cabinet at home. Hydrogen peroxide, right? So we'll dump hydrogen peroxide. We actually make hydrogen peroxide in our cells, right? Reactive forms of oxygen, and we're going to dump those on those cells or on those microbes. That toxic form, as the name implies, is toxic. It would hopefully do what to the microbes? It's going to kill them. Right? Hopefully it's going to kill them. Now, then we're going to expel by exocytosis any of the debris outside. Does this work against all microbes? Can we kill Shigella like this? No. Shigella knows how to escape the phagolysosome. He jumps out. Mycobacteria know how to stop the acidification of the lysosome, and therefore it also stops the lysosomes from binding. So you don't even get a phagolysosome. It grows inside of our macrophages. Okay. So we still have illness because unfortunately some microbes found a way around this. Some of them even can stop from being opsonized. Right? C3b doesn't bind to them. They have a capsule. It doesn't bind to that surface. Can't recognize it. So we can't even do the first step. We can't even recognize it to be able to phagocytose it. So although this is a pretty awesome system, right, there are microbes out there that have found their way around it or can escape from it. So the next set of cells that are granulocytes, right, which means they're filled with granules and also their names end with "phil," right? So we talked about these guys briefly last time: eosinophils, basophils, and mast cells. These guys don't do phagocytosis, right? They don't engulf. Instead, their granules, right, those vesicles that they have that contain digestive enzymes and other substances, actually go to the surface and get released. A lot of them are cytotoxic. In other words, they're going to kill neighboring cells, right? They have toxic effects on the cells that they dump their contents on. So basophils and mast cells we're going to talk about together because they're very similar. They both have blue to purple basic staining granules, and that's why they're referred to as basophils. Basophils are what you see in your blood. And remember, these are the least abundant. You'd be lucky if you see them unless you're experiencing some type of reaction, some type of immunological reaction. Your mast cells are similar to the basophils, but these guys are found in your tissues, right? So all throughout your tissues, when invaders get in, these guys can become activated. And what they're going to do is degranulate, right? They're going to release those toxic contents. One of the other substances they also have that's not toxic, but it's a communicator to our system, is histamine, right? So histamine, remember, is going to cause vasodilation of the blood vessels in that area and help contribute to inflammation. The problem is some people like myself who experience allergies, whose immune system is recognizing pollen or something like that as being foreign and causing the degranulation of mast cells and basophils. That causes inflammation, right? So you get the stuffy nose, right? Or people who inhale these type of things, different mediators get released and they get constriction of their airways. So it's kind of unpleasant. So most of us have heard of histamine because we've heard of what people take against it, right? They take antihistamine. Well, I got news for you. The way antihistamine works is it binds to the receptors that histamine binds to. It competes with histamine. So some people are like, I took my allergy medicine, but it's not working. Most people don't understand how allergy medicine works. You take it before exposure to the allergen. So if you're allergic to cat dander and you're going over to your friend's house who has a cat, when do you take your allergy medicine? At your house before you head to her house, yes? To get it in your system so that it can outcompete when the histamine is released. So when the histamine is then released by your system, it can't bind to those receptors to cause vasodilation because the antihistamine is bound to it, right? So should you still take it after you've already arrived at your friend's house? Yeah, because it's going to, once it gets into your system, it's going to help it to stop it from keep happening, right? But it's not going to take care of the initial inflammation that you're dealing with or response that you're dealing with. Right? That's a bad thing with these medications too being over the counter. I haven't read the labeling on them, but Unless you've gone to an allergist or a good doctor, most people don't even take them as they should, right, before exposure, if you can. So for my case, I'm allergic to like everything outside. I take it every morning before I leave the house, right? It's a ritual, me and my son, both of us. So how does degranulation happen? How do those cells know they need to dump these toxic contents or release histamine? Right? What's going to cause them to degranulate are things like C3A and C5A. Where does that come from? We've talked about this, haven't we? What are those? What's the C stand for? They're complement proteins, right? And they're pieces of complement proteins, right? They're the A piece, the A fragment of numbers 3 and 5. And remember, once this system is activated, and there are several things that can activate it, right? If it's the classical pathway, what's the first thing that's binding to the microbe and therefore interacting with complement and causing that cascade of events to go forward? What is a classical pathway? What's the first thing that binds? It's actually a product of your adaptive immune response. Antibodies. Where the alternative pathway, what complement protein is opsonizing and binding to the surface of the microbes? C. What one's the opsonin? We said it earlier. C3b. You need to know this one, right? Or we could have mannose binding lectin bind, right, by the lectin pathway. But then a cascade of events is going to happen, and we're always going to get the C3 convertase, right, that converts C3 into C3A and C3B. So we've got C3A right here. That's going to bind to those mast cells nearby and cause them to degranulate, and what's one of the things they're going to release? Other than cytotoxic substances, they're going to release histamine, right? And that's going to cause vasodilation in that area. Well, how does my body know that it doesn't like pollen? Well, because unfortunately my body has made antibodies specific against the pollens that I'm allergic to, like ragweed. We're going to talk about antibodies and adaptive, but I think it's good, and they do introduce it in your book here, to mention these antibodies. So antibodies, you produce 5 different types of antibodies. So another name for an antibody is an immunoglobulin. So we commonly abbreviate that IG. So this is actually an I. I hate this font. I probably should have changed it. It looks like an I to me. It's a capital I. Lowercase g. So that's immunoglobulin is what IG stands for, right? An antibody. When there's a letter after that, in this case E, it stands for one of the 5 classes of antibodies that we're going to talk about. So that's what they mean by IgE. So it's specifically the antibody class E. And I always tell my students, you shouldn't screw this one up, right? Because usually, before I got my allergies and everything else under control, I was always stuffy. And so I used to tell my students, just remember, E is for Erica. Erica hates her allergies, right? You won't ever forget that. It's pretty funny when you think about it, right? That it happens to be the letter E, and my name starts with the letter E. We will make these against parasites as well. And the interesting thing about these antibodies is, as we'll go over later, the structure of them, but they have antigen binding sites on them. That's why they look like they have 2 arms, and they're identical, just like your 2 arms are pretty much identical. These are actually what grab on to the pathogen. The interesting thing is this portion, this lower portion of the antibody, known as the FC region. So it's a large F and then a little C. You'll see that abbreviation. You'll see it coming up on another slide. Your mast cells and basophils have receptors specifically for that class of antibodies, FC portion. So what happens, unfortunately, in my body is that my mast cells get coated in IgE, specific to, say, ragweed pollen. So then when I inhale ragweed, it binds to the antibodies, and it causes these mast cells to degranulate. Within seconds, this reaction happens. So after one of the hurricanes, and I came back on campus and I walked into the building, I literally within seconds could not breathe. Right? Because I'm already sensitized. I already have these antibodies. They're already coating my mast cells. So it's an immediate type reaction that happens. Does that make sense to you guys? So for mast cells and basophils, it's IgE. For eosinophils, it's IgG. Right? So again, an antibody class, Ig, standing for immunoglobulin, the letter G in this case refers to that specific class, that specific group of antibodies. So those will bind to the surface of the eosinophils. Again, they have a receptor specifically for these antibodies. And when something that those antibodies recognize bind to it, the eosinophils will degranulate as well. And again, these contain toxic substances. But a little bit different than the basophils, hence why they stain differently, right? These don't contain histamine. They actually contain antihistamine. So that's it for our eosinophils, basophils, and mast cells. Now we're going to go on to our natural killer cells, commonly abbreviated NK. And these were discovered when they were doing research on tumor cells, right? So there's a little interesting block in your book about this. And they found that they could naturally kill tumor cells. So we've talked about cells that engulf bacteria, right, or microbes that are outside. We talked about ones that release their toxic substances on them, right, the eosinophils, the basophils, and mast cells. Well, what happens when something gets inside of our cells? Viruses do that all the time, right? What if we don't stop it from getting into our cells? What if it gets in? What if it's it's going to take over that cell? Now, we learned about interferon before. That's going to help us, right? But do we want to rely just on interferon and cells, telling neighboring cells, hey, bad guys here, get prepared and die if you have to stop it? Right? What if that virus gets in and deactivates that system? Do we want just one way to defend ourselves? No, right? It's redundancy, right? We have multiple systems to help defend us. So for intercellular parasites, bacteria that get inside, and definitely viruses, and even for when our own cells behave badly, right, when they become cancerous or tumorous cells, we want to be able to detect abnormal cells. And again, we're going to kill it. So if one of our cells is infected with a virus, a microbe, right, it's cancerous, we're going to kill that cell, right? Because one cell is not a big deal as compared to many cells, right? We're multicellular organisms. Thankfully, we can lose a few cells and it's not the end of the world. So again, they're not phagocytic, so they're not going to engulf, but they do make contact with the target cells. Once they make contact, right, they recognize that there's something wrong with that cell, then they're going to release two substances. They're going to release perforin. These actually perforate the membrane of the cell, and they allow granzymes, this one's a fun one, granzymes, to enter into the cell. These enzymes initiate a process known as apoptosis, or sometimes referred to as cell suicide. It's going to cause the death of that cell. Like I said, right? There's something wrong with that cell. We don't want it around anymore, so we're going to cause the death of it. So the important thing is this is something inside of one of our cells, right? Or even one of our own cells, right, being abnormal or a cancer cell. So there's different types of cell death. I just mentioned apoptosis, right? Programmed cell death. So the cells themselves are going to decrease in volume. They're going to go through physiological changes, and they're going to actually break up into little pieces. And phagocytic cells, like your resident macrophages, are going to engulf them and destroy them. The important thing about this process of cell death, this cell suicide process, it does not elicit inflammation. In this case, we don't necessarily need an inflammatory response. And when our cells, their lifespans have ended, all of them go through this process, whether it's been initiated by a natural killer cell, right, or by their own program. Necrosis, on the other hand, is a different type of cell death. This is where the cells themselves break up in such a way that they don't shrink into little pieces, right? They don't package themselves into safe little pieces. Instead, their membranes may be damaged and their contents actually even burst. Unfortunately, this is toxic to neighboring cells, and so you'll get death of cells that are nearby. And this is when you end up with, necrotic tissue, when you have a lot of dead cells. And this can be really bad because dead cells are food for invading pathogens, right? And dead cells can't help us because they can't do anything, right? They're dead. And depending on how many cells you have, if you get to the point where you've got tissue that's dead, you can't recover from that, right? That has to be removed. We're not that level of bionics or anything like that, right? We can't repair something that's dead. Right? So one of the diagrams I wanted you guys to fill in was this one. So how do our cells communicate is the main question and why we look at this diagram. So one of the ways is cytokines, right? Chemical messages. But do they actually physically interact with each other? Remember those neutrophils? How did they get into the area where we had inflammation? How did they get there? They just, jumped off the superhighway of the bloodstream and popped right into the tissue? The blood vessels dilate, but remember, why did those white blood cells physically roll along the surface of the blood vessels? Yeah, but why are they rolling? Why are they even interacting with that blood vessel?
Something pulls them, it connects to them and pulls them out.
Right, what's connecting? They have receptors, right? They have adhesion molecules, right, that the endothelial cells express, and the neutrophils have receptors for them. And so they're literally physically binding with each other, right? They have these receptors on their surface. So there's one receptor that natural killer cells help keep track of. Anyone know what that receptor is? You'll see that it's found on all of these cells. It's this little red symbol. But what did you guys write in for this one? This one you can look at your notes if you wrote it in. Who did their homework? Nobody? The little red one. On the endothelial cells, it's the only one in this diagram. But notice that it's on this one, it's on the natural killer cells, it's on neutrophils, it's on T cells, it's on B cells, it's on macrophages or dendritic cells. It's MHC class 1. And it's found on all your nucleated cells. So what is it? What is MHC class 1? It's an abbreviation for major histocompatibility complex. In this case, it's class 1. So we have two classes of this molecule. The first one is found on all your normal nucleated cells. But what is it? It's a receptor that will pick up proteins from inside your cells and present them on the surface of your cell. So what's the purpose of that? Well, think about it. When a virus gets in, it starts taking over our cell and making proteins for its coat, right? If MHC picks up that protein and puts it on the outside, now we can flag down our friends and say, hey, 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 look what's inside of me. It's a way of presenting antigen to the surface of the cell because these pathogens get inside. Also, when our cells become abnormal, they'll display abnormal proteins, right? Proteins that we're not making properly on the surface of our cells. There's another set of cells that will actually recognize those proteins. Natural killer cells aren't it. Natural killer cells are surveying this process. They're saying, are you presenting MHC class 1? Because think about this. If you don't display, say you're a virus, right? You get in, you're making your little viral proteins, and you stop that cell from presenting MHC class 1. They stop picking up your proteins and showing them on the surface. Is the immune system going to know the cell's infected? No, because it's inside, right? We've got to have a way of broadcasting this outside the cell. So if interferon's not released, that virus is inside, it's making copies, and we're not presenting that information on the surface, there's no way for our system to know that there's something wrong with that cell. Except that's the job of our natural killer cells. So virally infected cells will shut down this process of presenting, right? Because they don't want to be detected, right? They're like, they don't want us to know we're here, right? So we're going to, evolutionary wise, the ones that can shut down that system, it's been beneficial to them. Except for the fact that when the natural killer cell comes along, any one of your normal cells that's not presenting MHC class 1, because that's a normal behavior, right? To always be presenting MHC class 1, they're going to cause that cell to be killed. So virus-infected cells, this is common for them to sometimes not be presenting or not presenting enough MHC class 1. Or sometimes our cancer cells, tumor cells, when they start doing what they're not supposed to do, one of the things they forget to do is present MHC class 1. So this is a diagram showing what happens for a normal cell, right? So here's a normal cell, over here. The little red things, as we said, represent MHC class 1 receptor.
The second one's the normal.
This second cell right here is a normal cell. Normal unaffected cell. So here's that cell. It's presenting MHC class 1. You see all the little red receptors? It also produces an activation molecule on its surface that the natural killer cells have a receptor for, an activation receptor for. So these will bind up, but it won't get activated. It won't release its granules to kill this cell if CD94, so they've determined exactly what receptor is found on our natural killer cells, which I'm not going to make you guys memorize, actually binds to the MHC class 1. So this is an inhibitory signal, right? So even though the activation signal is binded, because it recognized that this cell is presenting MHC class 1, it's not going to kill the cell, right? So he checked out okay, we'll let him go. But say you have a virus-infected cell or a tumor cell, right? And it's not producing a lot of MHC. Notice there's just 2 of them over here. But there aren't any on this side of the cell. So it binds to the activation signal. There's nothing to bind, no MHC to bind to the CD94. No inhibitory signal gets sent. So that natural killer cell gets activated. It releases the perforin, right? The granzyme is going to go in, and it's going to cause that cell go through the process of apoptosis, programmed cell death, without inflammation. Okay. So it makes sense that all these cells, right, even our white blood cells, have MHC class 1 on their surface. So if something gets inside of them, they can alert the system. Natural killer cells recognize when there's no presentation happening. Now, phagocytic cells do what? They take stuff in from the outside, right? So that might be a bacteria that wasn't even going to come on the inside anyways, right? But instead we engulfed it. Do we need to present that information to the immune system? Yeah, we do. Right? Because sometimes we're going to need additional help. Because remember, sometimes phagocytosis doesn't achieve in the death. Or maybe it's overwhelming to the system and we need backup. We need to have some way of communicating this problem. So we have what are called antigen-presenting cells. And they have an additional class of MHC. They have MHC class 2. So notice macrophages, right, which engulf, and dendritic cells, which we haven't talked about yet, but we will engulf pathogens, digest them, and they will pick up those pieces and present them on the surface, but they will present it in MHC class 2. So will B cells. B cells will take in any antigen that they interact with, and they'll present on MHC class 2. But notice if they get infected, they also have MHC class 1. So they can present that information. Well, who are they presenting this to? Anyone actually read in their book? To a group of cells that we collectively refer to as lymphocytes. When we look at them under the microscope, we just see that they're a lymphocyte. But you have 3 different types of lymphocytes. Natural killers are actually in that classification of lymphocyte. But they are not recognizing specific antigen, right? They're just recognizing, hey, that cell's not presenting MHC class 1. That's not saying, oh, this is the flu virus, right? It doesn't know. Just knows there's something wrong with that cell. It's not doing what it's supposed to do. It needs to die. Your T cells, which are part of your adaptive immune response, or your T lymphocytes, have specifically on their surface what's called a T cell receptor. T cell receptors bind to MHC. They are what's going to now, remember MHC is holding that foreign protein, whether it be your own abnormal protein or a virus protein or intercellular bacteria like a mycobacteria protein. It's holding it out, right, to the immune system going, here, look, 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 this is what I got inside of me, help me. Who's going to help in this process? The T cells. T cells are what go around and inspect our cells. We have 2 types of T cells, though. And they're determined by this next set of markers that they have. So this next one that I wanted you guys to know is CD4 and CD8. So notice it says OR. So depending on what type of T cell you are either a CD4 T cell, where you have CD4 on your surface, or you're a CD8. And this is determined in your thymus, and that's why these are called T cells, right? Because they're educated in your thymus. So B cells, do they recognize pathogens, antigens? They do. And they have a special receptor on their surface known as the B cell receptor, but that shape looks familiar. What did I tell you that shape is? Antibodies. Antibodies. Their B cell receptor is actually an antibody. So this next slide is a very important slide as we go throughout adaptive, and we talk about these cell-to-cell interactions. So MHC class 1, we said that this receptor is found where? What cells on that wheel was the MHC class 1? Non-infected. It could be infected cells too, though, right? because we're going to pick it up. It's just sometimes the infected cells will shut down this expression. This is found on all your nucleated cells, your epithelial cells, right? All your white blood cells, macrophages, dendritic cells, natural killer cells. All normal, right? Behaving normal cells should present antigen via MHC class 1. And that's what they're doing with this molecule, right? They're picking up antigens from inside and they're presenting it outside. MHC class 2, though, is only found on a particular group of cells. We refer to these guys as antigen-presenting cells. So on that wheel, who were they? B cells had it, and who else? Macrophages and dendritic cells. So these are ones that are actually going to take antigen from the outside, process it, and present it outward. That's the difference for these guys. They actually actively take stuff in. Extracellular pathogens that would normally go inside of a cell. T cells have MHC class 1, right? But they're important in that they're going to talk to cells that are presenting MHC. Right? So their T cell receptor specifically, and this is the important part, specifically, right? So they know specifically what antigen you're looking at. But we have 2 classes of MHC, right? But do we have 2 classes of T cells? We do. We do. Right? Because this is different, right? You want to know, did that cell engulf the pathogen, or did that pathogen get inside of that cell? So for our antigen-presenting cells that have MHC class 2, they specifically communicate with CD4 T cells, and these T cells are commonly referred to as helper T cells, because they're going to do just that. They're going to help. Where our other class of T cells are our CD8. And it makes sense that these are cytotoxic because remember, MHC class 1 is for when stuff gets inside of us, right? When viruses and bacteria get inside, we present that antigen. And so then the cytotoxic T cell is going to come around, and what is it going to do to that cell that's infected with the virus or the intercellular bacteria? Why do we call them cytotoxic? What are they going to do to that infected cell? They're going to kill it. They're going to release cytokines and stuff similar, and cytotoxic substances similar to like natural killer cells. But the difference here is that T cell knows exactly what is infecting that cell, the specific pathogen. So B cells, right, have a B cell receptor. And as we saw, it's that shape, right? It's an actual antibody. The class for that one is IgM. So we've already mentioned 3 of the 5 classes of antibodies we're going to talk about later. I'm getting close on time, huh? I'm out of time. Okay. So let me mention the last one. Give me 1 minute. Can you do that? Okay. The last one is the last protective outcome that antibodies with natural killer cells can do. So again, this is a product of our adaptive immune system, specifically IgGs. We produce these antibodies, and think about this, when a virus gets inside of our cells, it starts, especially if it's an envelope virus, it starts putting its proteins in our membranes, right? So this isn't presenting by MHC class 1. This is actually putting its proteins in our membranes. Well, if we've seen this virus before, or if it's during this attack that we've produced antibodies, the antibodies themselves will bind to that cell. But again, antibodies are just opsonins, right? They tag for destruction. This particular type of antibody outcome, because it's natural killer cells that bind to that FC portion of that class of antibody, it's referred to as antibody-dependent cell-mediated toxicity. So we kill cells because of the antibody that the natural killer cell is recognizing. So again, not specific for the natural killer cell. The natural killer cell just says, oh, IgG coding a cell. I must kill that cell. Right? It's the antibody, though, that specifically recognizes what virus that cell is infected with. Okay? So we're going to start the next chapter, adaptive, on Wednesday. So start reading in that chapter. We're going to follow right straight through in the chapter like we've been doing.